Welcome, everyone. Good evening, or good morning, or good afternoon, or good middle of the night, whatever time it happens to be in your corner of the world. I'm really excited. We have a record-breaking 320, and the numbers keep climbing, participants here today. And I think that number really speaks volumes about just how special and exciting and important this special edition of the ICSA virtual seminar series is for us. And of course, we have here with us a scientist who needs absolutely no introduction in our field, and that's none other than Dr. Leonard Hayflick. So Dr. Hayflick has been, I think I speak for all of us when I say, an inspiration for each and every one of us at some point along our career, from our infancy as scientists, kind of reading about the discovery of the Hayflick limit, through our trials and tribulations of this thing we now call senescence biology. And I just think it's an absolutely incredible privilege that we have to have this man who basically is the raison d'etre of this entire society that we have, who made this paradigm shifting discovery and found that actually individual cells have a lifespan, individual cells can age. And now, actually last month, the seminal Hayflick and Moorhead paper turned 60 years old. And in these 60 years, we've had this incredible opportunity to watch the field grow. Well, not me, because I haven't been here for the whole time, but watch the field grow into this scientific behemoth that touches every corner of the world, that touches almost every biological journal, and has massive implications for health, for disease, and for this profoundly philosophical question of how we age. So, as I said, Dr. Leonard Hayflick really needs no introduction. He has more accolades than I could mention. I could spend the whole seminar just talking about them. But just to say that beyond his role as a scientist, he's also served as a consultant on many kind of colossal government organizations like the NCI, in biotech as well with Genentech, and he's contributed to many, many different fields beyond just cellular senescence, including mycoplasmology, vaccine development, and the use of cells for the development of biologics. So Dr. Hayflick, thank you so much for being here today. Thank you so much for sharing your experiences, and thank you for the inspiration that you've given generations of scientists, and I'm sure many more generations to come. And the floor is all yours.
Well, thank you very much. I want to thank all of you for this invitation. I have to confess that I didn't know anything about your organization until a few weeks ago, a month or two ago, when I accidentally ran across it in an internet search. So I was really astonished to learn that there was in existence this organization. But it really represents a major advance in understanding this area that, as I'll explain later, initially was ridiculed. But so in the lecture that I intend to give, I want to encourage you to read text on slides more than you have in other lectures. And I'm doing this because of the need to reduce the length of verbally describing the historical events that occurred in my discovery of cell senescence. This will allow for a more succinct presentation of the events and a better guarantee that they will be explained in chronological order. As part of the history, I'll show some slides that were made about 60 years ago when I first lectured on my findings, just because they might be amusing and of some historical interest. To understand what follows, you really need to have a 2-minute course in cell culture technology. Those who know this technology can now take a very early 2-minute break, but I will give this introduction to cell culture technology in about 90 seconds. So you take a scrap of tissue from some donor source, animal or human even, and the cells look like bricks in a wall. You dissolve the mortar with an enzyme, releasing the individual cells. You place them in a square vessel with growth medium. The square vessel is placed on its side, so you have more area. And if they're human cells, of course, you incubate them at 37 degrees Celsius. The cells slowly settle down onto the floor of the square bottle in a few hours, and in about a day or so, they begin to divide, and over the next few days, they divide such that they cover the entire floor of the vessel, and then they stop dividing. If you want more cells, and that is what's of interest here, the growth media is poured off, the enzyme is added, it separates the cells from each other and from the floor of the flask, and then the suspension is divided in two equal parts for convenience of simplicity in this case, and each half is introduced into a new vessel with fresh growth medium and incubated. And you can do this serially. It's called a 1-to-2 split in lab technology, or a subcultivation. So the process can then be repeated many times to obtain millions and millions of more cells. So with that brief course in cell culture technology, let me begin. I'm going to make a presentation, as I've been requested to do, of a brief history of the scientific and legal consequences of the discovery that normal cells are mortal and age, and that cancer cells are immortal and do not age. And I'm beginning here from 1956 to 1902. So I received my doctorate from the University of Pennsylvania in 56 and accepted a postdoc in the laboratory of Charles M. Pomerat, who was then one of the leading cell culturists in the world. I'm going to let you read some of these short paragraphs, as I indicated earlier, in order to save some time. I'll interject some thoughts as we go along. So this is just an additional observation that I made actually at the same time I was looking at cultured normal human cells. I thought this was an interesting aside. The last paragraph is a key paragraph because it left me with opportunity to do whatever research I wanted to do without the burden of applying for grants. And that's the reason I accepted this service laboratory position. So now we're moving to 1961. At that time, this was a situation in cancer research. Yes, the conversion of normal human cells or animal cells to cancer cells bore the name transformation. I understand that in the past 60 years, that term has been used for other meanings, but this was the initial meaning, and this is the meaning I will use it as. And again, the last paragraph from this slide is essential because the randomness of the phone calls announcing the availability of fetal tissue became a key part of my discovery, as you'll soon learn. I hope I'm not going too fast for you here. And as I've indicated in this heading here, one of the surprise findings that I'll describe momentarily was that normal cells have a memory. They certainly don't have a brain, but they do remember, and that was a very strange discovery. And the discovery, that finding was based on the fact that we could preserve cells at sub-zero temperatures. And because the normal human fibroblasts derived from fetal tissue underwent about 50 population doublings, and I'll explain that in a moment, it allowed me to preserve cells from the first to the 40th or 50th doubling. And the term PDL was first used by me to indicate population doubling level. It's not cell doubling level, it's population doubling level based on the first release of cells from intact tissue being the initial population. And there's where the memory was found. So that was rather striking, but not clearly understood, of course, at that time. And the discovery itself, about which I know many of you are interested, occurred as follows. And as I indicated earlier, this fetal tissue was received at random times whenever there was a surgical abortion that allowed me to obtain tissue, and that was random. And as I say here, it was not unusual to find one bottle of cells not dividing or behaving erratically because people didn't understand the technology at that time. And so, as other cultures in this series that I just described also showed cells that had stopped dividing as I subcultured them, that became more interesting, because if one culture failed, you just dismiss that. But if you find others also failing at about the same population doubling level, then this is what happens. It was not a eureka moment, it was a that's really funny moment. Why is this happening? Why are the cultures that were showing cells luxuriating as they replicated now stop dividing after 40 to 50 population doublings. So that's why I call it my that's funny moment. And that, of course, excited my curiosity. And I guess that's what all scientists have to be aware of, to be curious about something unusual they see. And so we tried to find out what the cause was, because at that time, everybody believed that if your culture stopped dividing, it was because of something you did wrong, or the cultures became contaminated, or the glassware, plastic wasn't used at that time, or the glassware was somehow not cleaned properly. Fortunately, Paul Moorhead, my colleague, a cytogeneticist, found a discovery that was made at that time, which was very fortunate, that we could distinguish male from female cells by being able to stain the cells and seeing the inactive X chromosome in female cells. It was called the bar body. So this is the experiment that we did in the last paragraph here. Very critical experiment. So the mixture of young female and older male cells was we kept subcultivating along with the unmixed cultures of each. And then we decided to look at the mixture after about 10 more doublings, and only female cells were in the mixture. And you see what happened to controls at that same time. So the conclusion was clear. The probability that some technical error or media fault and it proved, at least to me and to my colleague Paul Moorhead, that we had not made a mistake, as many people said who didn't want to believe what we found. And we were, I was persuaded to believe that we had not made a mistake. And from that occurred these other observations. The 60-year-old dogma that was established at the founding of cell culture in the early 1900s, that dogma said that when you put cells in culture, they all, regardless of their source, at least those that can divide, have an infinite capacity to replicate. And if they're lost, or if they fail to replicate, you did something wrong. And that dogma was held by the leaders in the field. And this was the key finding, of course. Cancer cells are immortal, normal cells are immortal. And that would never have been known until this time, because the dogma was that all cells are immortal. This was the status of the field of aging at that time. The belief was that aging was caused by extracellular phenomena. But I reversed that belief to the notion that aging has an intracellular etiology. And this sentence added more weight to my belief. So I concentrated on one cell strain called yI38 because I found that all of the strains that I developed from other fetuses behaved similarly, almost identically. So this is one of the 60-year-old slides showing over the period of a year what happens with normal human cells in culture. There's I call it phase one, which was the primary culture, that is the culture that underwent no subcultivations. And phase two is where they've been subcultured over many months. And phase three is the period when they stop dividing. And at that time, the term cell alteration was also used synonymously with cell transformation. And it was known that that happened. It never happened with my cell strains like yI38 or any other normal human cell strain, to the best of my knowledge. And this is the appearance of the cells as I photographed them at that time, stained, of course. On the left are the cells as they are replicating in phases 1 and 2. You see a polar view of mitosis in the lower third of the slide with chromosome stained. And on the right, phase 3, with the cells showing completely different morphology. They're spread out, flattened, etc. And here is the margin of two cell states. What you see here is a culture of human amnion cells, and a transformation has occurred. And here are arrows pointing to the periphery of the cells to the north of the arrows that are the transformed immortal cell population. And the cells below the arrows are the normal untransformed amnion cells. This amnion came from the birth of my daughter, Susan. So I should give thanks for technical assistance to my wife, and I'll do that. This cell line is called WISH. Some of you may have heard of it. It's not widely used anymore. It used to be used. So here is a transformation of normal yI38 with the oncogenic virus SV40, showing the difference in morphology. The cells on the left, of course, are mortal. The cells on the right are immortal as a result of the transformation. So with all of this information, I reported in my papers that cells in culture have three general states. The first are primary cells with the characteristics that you see here. The second are cell strains. It's the second class of cultured cells. And finally, cell lines. Many of you will know cell lines, most likely from HeLa. And those are the properties of cell lines. So it was time to publish. And this is the reason I chose the Journal of Experimental Medicine, which was a Cadillac of journals at that time. Some of you may know of the work of Alexis Carell, who received enormous worldwide notoriety for this allegedly immortal cultured chick heart cell strain, which we are certain he either did not know why his culture seemed to be immortal because he was seeding it with live chick cells at almost every feeding, or he and more likely his technicians who were culturing these cells were afraid to tell him the truth. That argument has been discussed in print. I won't go into the details now. Of course, this was a great disappointment. This was the first really what we thought was our most important paper, and it was rejected. This is a little summary of the work of Alexis Carell. During those many years, his culture was fed with chick embryo extract, which he thought was made by removing all of the living cells in the extract. That turned out not to be the case. So he was seeding his immortal chick heart cells with fresh chick fibroblasts at each feeding. Carell was notorious for his for encouraging the press to advertise his research, and his hiring of Charles Lindbergh was the ultimate in that pursuit. Here is the letter that rejected my paper, signed by Peyton Rouse. You can't read this easily, but I'll quote from it on this next slide. Here are the key quotes. This is the first one that's key that many of you will be interested in. That's one of the reasons the paper was rejected. Second reason was this. Of course, we thought that the evidence in our paper torpedoed this idea, but even winners of Nobel prizes sometimes make mistakes. So this is the first page of the paper that I think you indicated earlier you earlier many of you have read. It's been cited more than 8,000 times up until today. Then I published a second paper. This is a copy of the first page. I don't have a photo of it, but I'm sure many of you are familiar with it. This is the first couple of paragraphs. I won't read them because I understand many of you are familiar with this second paper. So the first publication described differences between mortal and immortal cells in vitro and in vivo. And this is essential because many people who have never read the paper but referred to it don't realize that this equation that you see here was printed in the second paper. And this shows the equivalence that cell lines have, that is immortal cell populations, they have to transplantable tumors, as cell strains have to normal somatic tissue. And here you see the conditions of where these cells occur. Cell lines are heteroploid or aneuploid, as are transplantable tumors. Cell strains, if derived from normal tissue, are diploid. These are second properties. Third properties of this equation. And this is all copied directly from my second paper. And that was a key distinction that ultimately was appreciated. As I indicated, that's the number of citations for that one. This is the number of citations for the second paper. So this is important, as I indicated at the outset. None of my work so far described, and most described subsequently, was not done with any NIH grant or any other research grant. It was all bootleg material that would otherwise be discarded. Any of you who have worked in the cell culture laboratory will know how much is discarded in the way of media and cultured cells. There is a NCI grant credited on my two papers, but those grants were made to the Institute as a whole, not to me, for overhead support. And separate from that, when the demand for WI38 cultures rose to a point where I could not meet the demand, the NIH, who had many people working with my cells, offered to give me this contract to store, manage, and distribute the cultures because of this unique situation of having hundreds of people worldwide asking me for cultures of WI38. Most of the reasons for requesting them were from virologists worldwide, because I had shown in my first paper their exquisite sensitivity to all then known human viruses. This is also important for the first report of the changes that occur in normal cells as they reach phase 3 and are comparable to the changes that occur in the cells of older people. So it was obvious if cells could remember what population doubling level they had reached at any time, their memory, they must have a memory. Obviously, it's not a brain. It must be some mechanism, and clearly I hypothesized it probably has something to do with DNA replication. And we want to pursue that idea. And with my student, Woodring Wright, we found that the counting mechanism was not unsurprising in the nucleus. And I called the mechanism, about which I knew nothing in 1975, a replicometer. And about 10 years later, this discovery, major discovery, occurred. The discovery was based on the earlier findings in '84 by Grider and Blackburn. But here was the key observation made in 1990. So this major discovery occurred because of an accident. So I think it's becoming clear that a lot of the things that occurred with me and in this field are the results of accidents. And that's not an unusual situation in science. Accidental meetings or fortuitous occurrences commonly give rise to major discoveries. So this was a major discovery made by Grider and Cal Harley and Futcher. And you all are aware of this, I'm sure. And the final paragraph is also key. So now we have a molecular explanation for my findings at the phenomenological level. This is another ancient slide, about 40 years old, I think, after the discovery of telomere attrition in the culture of normal cells and the expression of telomerase in transformed cells or cell lines. So I just showed as a matter of an old historical slide. So other things also occurred. This was an interesting one. Because by this time, in 1973, WI38 had been used so widely throughout the world and had been characterized in respect to its genetics, its biochemistry, and many other aspects of its biology, that it was easily selected by NASA to be flown on Skylab 3. No other culture then, and I think even to this day, has been characterized to the extent that WI38 has. For those of you who are interested in the reference for this work, there it is. This is simply me at a very youthful stage in my life, showing the string of chambers that were kept on Skylab 3 and photographed for study. Now this is another aside. Some of you may not know this, but in the early days, it was not possible to see cultured cells in flasks because they're too thick. So the next time you look through an inverted microscope in a tissue culture laboratory, you'll soon see a picture of its grandfather, maybe its great-great-great-grandfather. There it is. So now let's move just for a couple of slides to the use of WI38 for the production of human virus vaccines. In my first paper, I noted, as I indicated earlier, the exquisite sensitivity of human diploid cell strains to human viruses. And most importantly, that my cell strain, like WI38, did not contain contaminating viruses as primary monkey kidney cells did at that time. These cells were used for the production of the world's poliomyelitis vaccines, both the Salk and Sabin varieties. So the need for a safe and virus contaminated free cell substrate to grow human vaccines on became resolved by the use of WI38. Although that took about 10 years of political efforts, I won't go into those details. Resistance to change is a common phenomenon in science, as I'm sure many of you know. These are some of the major advantages of WI38 over monkey kidney cells, or any other primary cell for that matter. It's important to know that I gifted the WI38 cultures to the scientists at the Wistar Institute who developed the rabies and rubella virus vaccines that have been used ever since worldwide. So they received, I gifted to them the enabling technology that unfortunately does not seem to be appreciated. And I make that statement again here. So here's just an example. This is one of the first polio vaccines produced by Pfizer using WI38, as you see indicated here. This is the first paper that I wrote with Plotkin and Norton and Poprasky in 1962 to prove the efficacy and safety of polio vaccine produced in WI38. This was part of the politics of the day. So this is a summary of the benefits that WI-38 has had worldwide. These are the vaccines that have been produced using WI-38 that I gifted to not only to all of the scientists who requested it, but to the vaccine manufacturers worldwide. So this is the evidence here. This is the reference for this data. I was asked to testify before the Senate Committee on Government Operations because of some concerns about the behavior of the Division of Biologic Standards, which at that time was not a part of the FDA, but was part of the NIH, and they had supervision over the licensing of vaccines in the United States. I couldn't get a copy of this report, so I've excerpted the key parts of it for you. I argue that the Division of Biologic Standards should be removed from the NIH and moved to the FDA, and my reasoning was as follows. I won't comment other than having you read these short statements. So my testimony led to the removal of this Division of Biologic Standards from the NIH to what I thought was more appropriate, and that is to the FDA. And some of you may know that it is now that organization has undergone several name changes. This is the latest iteration. Well, I was, many of my colleagues at NIH who worked at DBS were very unfriendly as a result of this because of the reason given in this last paragraph. They were known to be researchers at the NIH, and now they became known as controllers at the FDA. And then, because of this antipathy, when I was at a meeting in Israel, they confiscated the WI-38 ampules from my lab in my absence. I was given a grant at Stanford to do research on cellular aging, and I was awarded a contract to distribute starter cultures to WI-38 only to research on aging. So now I had to, as a scientific obligation, to continue to send cultures to hundreds of scientists and vaccine manufacturers worldwide, for which I had no way to pay the costs. The costs, as you can imagine, were considerable. So I patterned my charge for preparation and mailing cultures to the same identical costs that the American Type Culture Collection charged for the same cultures of WI38, but at higher doubling levels. Most people wanted them at early doubling levels. And over a 6-year period, the prices at ICSA were these, and I charged similarly. I opened a bank account to receive these funds to await a decision as to their owner. I had no intention of believing that I was the sole owner of these funds. But then it erupted, as you read here. This was a unique situation in law because of the property of self-duplicating cells. Do you own the vials originally and all of the subsequent cultures made from the replication of cells from that initial vial? What does an owner of a cell line or strain own? That never was settled. And as far as the funds are concerned, I didn't know who owned the funds either. So I did this. The brilliant lawyer turned out to be an accountant. Of course, he didn't understand the biology and he didn't understand the law. No lawyer did at the time. But in any case, he accused me of not using that word thief, but he accused me of selling WI38. Of course, I was not selling WI38. I was simply requesting reimbursement for the cost of sending the cultures to people, and then putting the money in an account, separate bank account, to wait decision as to who owned the money. But that went over the head of the accountant. I will be finished in a couple of minutes. I know I'm a bit over time. Okay, this is the out-of-court settlement that eventually happened. And I will be ending this in 1 or 2 more slides. These are the reasons the government had to come to me and plead for an out-of-court settlement. Circumstances worldwide had changed enormously to benefit me and not the government, especially number 2. These are some of the reasons why the government found itself in a corner. So here's the conclusion. It was costly, but it was worth it because it settled a major problem. And this is important to every biologist. It established this concept that biologists have intellectual property rights. And in fact, 83 scientists from around the world agreed with my position and published a letter that was published in Science by Dr. Bernard Strahler, castigating the NIH and FDA's position in respect to me. And I will show you the list of signatures, but I don't expect you to read them all. I'll just show them to you. If you want to read them all, you can go to the original publication in Science. The last paragraph pretty much sums up what the signatories said. This is the article reference. So as I said, I'm not going to read these names. You can just go through them. Maybe you'll recognize one or two, but of course, these folks signed this 30 or actually more than 40 years ago. Many of them are retired. Many of them are names you may not remember, but most of them are well-known scientists in various disciplines worldwide. This is a final remark. I won a grant, an R01 research grant. This is what the disposition of that grant was. Vindictiveness on the part of the federal government. This is the key statement. A memo was sent to the director of Department of Health, Education, and Welfare, the organization under which is the NIH and the FDA. And the memo read as follows. The conclusion, of course, is obvious. These are some of the lessons learned. You'll be surprised to learn that there is no organization in the United States, and I doubt in any other country, that has the word aging in its title that is supporting research on the etiology of aging, which is the greatest risk factor for all age-associated diseases, and in fact for the leading causes of death. One of the reasons for this neglect is what I have here in this paragraph, what does research on aging mean? It can mean anything. It rarely means fundamental biology of aging. And this is my conclusion, for which I will defend to this day. So thank you very much. I appreciate your attention, and I'm sorry to have burdened you with so much reading. Thank you.
Thank you so much. That was an absolute pleasure. And thank you for the insights and the stories and the experiences that you've shared with us. It was really amazing. We'd now like to open up the floor for question and answers. I see that we already have a couple of questions that have come into the chat. If you'd like, you can also go ahead and raise your hand so that you can ask your question directly. If it's okay, I'd like to start with a question myself. So I think that I found a lot of recurring themes in your talk, kind of about the obstacles and the difficulties in academic research and the injustices in academic research. And it struck me a lot to see actually how similar they are to a lot of the challenges that we face today. And I wanted to ask you, in your opinion, if you think that these kinds of problems, how they've evolved over time, whether they're improving, staying the same, getting worse, and kind of give a little bit of insight on that.
Well, of course, I'd like to think that they're improving, but I have no direct evidence for whether they're improving or being disproven. But it's important to understand that scientists are people. And since you and our viewers are all scientists, they understand that. The public may not, lawyers may not understand that, and that we all have feelings, and we all seek the truth. That is something perhaps where we differ from many other disciplines, certainly in politics. But the search for truth, and that is not an easy search, and evidence for the truth is something that's not easily obtainable. I wish I could answer your question with more clarity, but I really have no basis to believe that things have improved, because I could argue that human nature has not improved, whatever definition you might want to put on those two words.
It's a very thoughtful and interesting reply. Thank you. We also have a question that was sent in to us from Daniel Munoz Espin, and he said, what are your 3 favorite discoveries or papers about senescence over the years, and which ones surprised you the most? And he added, thank you very much, Master, for inspiring all of us. We are forever grateful.
Well, the answer to that question really depends on what the questioner means by senescence. I didn't have time to go into the arguments and research in the field of aging generally, but I think the best way of answering that in respect to major observations in the field of aging that have been made in the past few decades would be to point out that there is a huge difference, and this difference is not appreciated in my judgment, between aging and the determinants of longevity. Almost all that is done in research today, allegedly in the field of aging, is done in the field of longevity determination. They are two opposite sides of a coin. Aging has to do with catabolic processes, that is, processes that fail for one reason or another, either slowly or rapidly. Longevity determination is an anabolic process. That is, all of the processes in a living entity that serve to keep that entity alive. And those pathways, those biological pathways, are known to be capable of manipulation. That's been known for over 150 years, where manipulation of temperature, crowding, food or lack thereof, can control the longevity of many animals. And that has been known, as I say, for 150 years. What has not been known or done is to tamper with the process of aging. That is, with the catabolic process, and that's what is missing in the entire field of aging: attention to the landscape of molecules in old cells as they are distinguished from the behavior of molecules in young cells, so that that landscape will hopefully provide insight into the reasons why the leading causes of death are associated with aged or old cells. That is, cells at the end of a lineage or cells that haven't divided at all or very little. And that is I think, an unsatisfactory answer to the questioner, but it does have some peripheral and very significant importance in my judgment.
That's great, thank you. Perhaps somewhat related, we have a question, well, from actually someone who has anonymized their name, so we don't know whom it's from. But they've asked, do you think that senescent cells, or I suppose these age cells that accumulate during aging, have a physiological benefit?
Well, first of all, I have not done any experiments with senescent cells in vivo. I can only depend on what I've read. And I'm trying to recall, without being successful, any papers that I've read where authors have suggested some benefit for the presence of aged cells. There might be some benefit, certainly, but I think that has yet to be discovered and proven.
Okay. Great. We also have another question from Atamana, who said, what are your thoughts on agents that extend the PDL beyond the 50 PDL limit?
Sorry, could you say that a little slower, please?
Of course. What are your thoughts on agents that can extend the population doublings, the PDL, beyond the 50 PDL limit that we normally see in cell culture?
Well, because we think we know the mechanism for the cessation of the doublings, that is, the attrition of telomeres, one would have to speculate on the likelihood of interfering with the telomere attrition. I don't know of anyone who has succeeded in doing that, but one of the arguments that has been made, a very common argument, is that overcoming the limit on normal cell replication can only be done, at least as far as we know today, by the cell's expression of telomerase. And that, of course, provides the cells to obtain properties of cancer cells. Something, of course, that's not desired. So I think we're at an impasse, at least in respect to where our research understanding is today, in any effort to try to extend the number of population doublings of normal cells without the intervention of telomerase. I don't know of any examples of that being reported.
Great, thank you. We also have another question from David, who says, there is now an accumulation of data indicating that some subpopulations of cells induced to senesce, for example, by chemotherapy or radiation, can escape and regain their proliferative capacity. This does go against the long-established paradigm that senescence is irreversible. What would be your thoughts on this?
Well, if you're defining senescence as the limit on population doublings of normal cells, then that clearly has occurred by telomerase expression. And maybe the agencies that the questioner described, like radiation, et cetera, chemotherapy, can induce the expression of telomerase in normal cells and have them pass the limit that I described. So I'm not particularly surprised at that result because it's been known for years.
And just one comment, Taranjeet Singh Rai wrote, low oxygen significantly extends PDL. I managed to go up to 90 just by careful culture and low oxygen genes development 2014 Rai et al.
Well, let me make one statement about experiments like that. Rarely have people who report an increase in population doublings by some intervention like that just described have determined that the cells that have reached that new level are normal. There is the great likelihood, and at least in those cases that I have personally examined, that likelihood has always occurred. The cells become transformed, and the people who see that event occurring do not establish the key properties of normal cells, namely their chromosome situation, that is, whether they're chromosomally normal, whether they behave as normal cells when inoculated into immune-compromised laboratory animals, whether they are incapable of growing in suspension. There are several other attributes of normal cells that we've known for decades, but those are rarely information, which has been known since my publication 60 years ago, is often ignored. You have to establish that the cells are normal if they've exceeded whatever limit you have discovered occur when the cells are absolutely known to be normal.
Absolutely. Okay, so we're going to maybe just do a couple more and then we'll let you go because we've taken a lot of your time already. But Adam asks, what do you feel is the reason behind the resistance to funding research on fundamental aging processes?
Well, that's an excellent question. The resistance I touched upon in a trivial way in my presentation at the end, but there is a vested interest, it's unconsciously vested, in the study of diseases. Aging is not a disease. I have published 6 reasons why aging is not a disease. I won't go into the 6 reasons now. But even if it wasn't, it turns out that most people don't understand that it is the condition of an old cell versus a young cell that provides the molecular landscape that increases vulnerability for age-associated diseases as leading causes of death in all developed countries. When it comes to the differences between the molecular landscape of old cells as opposed to young cells. That has not been investigated, despite the fact that within the past 5 or 10 years, we have the technology now to look at single cells and the molecules of single cells. Take cryo-EM, for example. There are many other technologies that are reported almost bi-monthly now in respect to new technologies to examine single cells or multiple numbers of cells at the molecular level. Let's find out what the differences are between old cells and young cells, because the leading causes of death are age-related. Why is that so? And why is it ignored? The other reason is political. When you go to politicians and governments and seek support for research on Alzheimer's disease, cancer, cardiovascular disease, or stroke, they know what those diseases are because they either suffer from one of them themselves or know friends or family who have. But when you tell them you want money for research on aging, they don't understand the reason for that. And the people who pose the requests for funds to politicians do not adequately explain that the aging process and the landscape of molecules found in old cells could in fact provide an answer to the question of why aging increases vulnerability to the leading causes of death. And that could be a single cause. It could be a single cause in the behavior of molecules in old cells that provide the conditions for increased vulnerability to pathology. And that is totally ignored because of the political reasons that people who go to politicians for funding cannot explain adequately why aging is so important to understanding this. And I have put my neck out and cited the statement that the leading cause of X is aging by every leader of every institution in the United States who heads an organization with aging or cancer or any other major cause of death in its title. I've just published that paper. I put the reference here. I'll be happy to repeat it later on if you wish to have it. So that the leadership knows that the major risk factor for the leading causes of death is aging, but they do nothing about it. We've got to educate these people.
So I think just maybe this can be our last question, but I think it's very kind of fitting with what you just mentioned. So several people have asked what your opinion on the therapeutic potential of senolytics is. So these types of drugs that can specifically eliminate senescent cells in vivo. So people would like to hear your thoughts on that.
Well, despite the fact that what appears to be an entire industry of startups and established pharmaceutical companies in a quest to get rid of senolytic cells in the hope that that will somehow interfere with the aging process, I'm very pessimistic that that would have any likelihood for success. First of all, ridding an animal's anatomy from aged cells, even if there is a single class of those cells, will allow for the expression of behavior of cells that are not senolytic. And furthermore, cells that are not senolytic could become senolytic after the first effort to remove them has been successful, even if that has ever occurred in humans. So again, I'm extremely pessimistic about any effort to interfere with a process that is, in my judgment, not biological. Aging is a problem in physics and not biology. Aging is a universal phenomenon based on the second law of thermodynamics. And it is also important to point out that aging is a phenomenon that occurs only in humans and in the animals that we choose to protect, like laboratory animals, domestic animals, farm animals, zoo animals. Aging does not occur on any scale that comes close to the scale that occurs in humans, amongst feral animals, certainly not among the largest biomass on the planet, and those are marine creatures. You do not find aging occurring in marine features by whatever criteria you want to use. Now, that's another serious problem in this field, establishing criteria to understand what aging is. But I will stand by the statement that aging is a problem of human civilization only. In respect to our success, if you can define it as success, some people will, some won't, in having eliminated causes of death that occur after reproductive success. And that's what we have done with our advances in medical sciences, in treatment of diseases, in public health measures, and in all the caregiving processes that we know about. So that aging is a unique problem created by humans, where we have allowed ourselves to live beyond the time when we should have been killed by disease, accident, or predation.
Absolutely. And maybe just one last question. This comes from Madeline, because I think it's quite a nice question. She says, your discovery of senescence challenged dogma at the time. What would be your advice to young scientists today if they are challenging what is currently known?
That is also an excellent question. I can only tell you, based on my experience, that because the experiments that I did in collaboration with Dr. Moorhead, that demonstrated to us that we were not dealing with an error or some mistake that we may have made in this finding of limited replicative capacity. So you must have faith in your experiments, and hopefully the experiments are decisive. And we felt that the experiment that I described, there are many others, but the one that I described came close to being absolutely decisive. And then you have to have the courage to stick by your observation in the face of ridicule, which I experienced for many years. My work was not accepted immediately upon publication. It's hard to measure, but I would say at least 10 years before people began to accept this notion. Other than that, I can't provide any advice that would be more significant. But thank you for that question. It's a very good one.
And thank you for that insight. I think that we all really appreciate it. Yeah.
Well, I just didn't want to end before thanking you and your colleagues for arranging this, and certainly for the establishment of the organization of which you represent a large student body. It's good to know that one's research reaches such a level of interest. And I wish you all good luck, and I hope to remain as a participant in this organization in whatever capacity you might find me to be of use. But thank you again, and good luck to you all.
Okay, with that, I think that we can wrap it up. Thank you so much for being so generous with your time and your experiences. and for sharing these hours with us. And thank you to everyone who joined us here today. Thank you so much. Have a great day.
You
Bye-bye,
too.
everyone.
Bye-bye. Bye-bye, everybody.